Yes.
How are you?
I'm good, doctor.
Good. Good to see you. So I think we start by 11:10.
We can start now.
We can start now? Okay.
Students, we are entering into the second session for the day. We have with us Dr. Gopalji Jha. He is a staff scientist 5 at National Institute of Plant Genome Research, New Delhi, India. Previously, he served as a scientist from 2007 to 13 at CSAR, Institute of Himalayan Bioresource Technology, Palampur, Himachal Pradesh. And he is also a visiting scientist from 2010 to 11 at the Sainsbury Laboratory, John Innes Center, Norwich, UK. He has several awards like and honors to his credit, like Swarna Jayanti Fellowship from DST, INSA Fellowship Medal for Young Scientist 2010, NASI National Academy of Science India Platinum Jubilee Young Scientist Award 2007, BIIS Fellowship from DST, Government of India, and Gold Medal, Guru Nanak Dev University, Amritsar, Punjab. He had his PhD from CSAR, CCMB, Hyderabad, and Masters in Biotechnology from Guru Nanak Dev University, for which he got a gold medal. So with this short introduction, I invite Dr. Gopalji to deliver his lecture. The floor is yours, sir.
So, thank you, Dr. Umar, for the kind introduction and giving me this opportunity to share my work and take to you through how to control diseases in a sustainable manner. So I will go slowly, and I request all of you, if you have any queries, you can either intervene me at that point or at the end of it, we'll be able to discuss about it. Okay. So as you might have seen, the title of my presentation is Novel Approaches for Plant Disease Control and Growth Promotion. So you all know that we really grow enough foods. Okay, so that is our take-home message, and you might be seeing in the newspaper that we have surplus food, we produce this many grains and things like that. But still, in spite of all the facts, still millions of people go hungry. And this is World Health Organization report that in spite of all the efforts, still millions of people we don't able to properly feed them. And further, we are expected to produce 60% more food by the year 2050. So this is very challenging. In spite of all efforts, still there are millions of people which go and hungry, and we are expected to have around 50%, 60% more food production. So how to sustain them? But on the top of it, we lose lots of food production due to various diseases and pests. So the estimate says that 1/3 of the total food produced per year, that is around 1.3 billion tons of grains, is lost or wasted globally. And amongst that, around 20 to 40% of the total loss is due to various diseases. And I've just put some of the pictures of some of the diseases which you might have come across. You might have seen oranges rotting. This is called molding orange. You can see the rice being infected with fungal pathogen. There is one fungal pathogen called seed blight. Another one disease is called rice blast. And you might have seen apple infected. So this is just a few pictures to tell you the importance of the plant diseases, but there are hundreds and hundreds of diseases which adversely affect the crop production. So how to control them? So people use agrochemicals as fungicides or pesticides to spray on plants to protect it. So here I am showing you an apple orchard where a truck is spreading the fungicides to control a fungal disease called apple scab disease. And you can see in one growing season of apple, around 12 such sprays occurs in the orchard. So imagine an apple which is almost coated with a fungicides. And I'm sure you must have read that fungicides have residual harmful effects on the health of the consumers as well as the environment. So this is really alarming situation, that though we have control measures, but that is not sustainable and that adversely affects our health as well as the environment. So how to control the disease? On the top of it, there are other challenges which is adversely affecting the crop production. I told you that we have to continuously enhance crop production, but climate change, degrading soil health, emergence of newer diseases, pests are posing further challenge to the crop cultivation. So question is how to sustain agriculture with minimal use of harmful agrochemicals. So today I'm going to take you through and emphasize that if we can harness plant-associated microbes, you all know microbes are the tiny organisms that live in association with plants and freely living in the environment. Those plant-associated microbes, if we can properly understand them, those can be helpful for us to have sustainable agriculture in future. So the take-home message is that the opportunity lies with harnessing plant-associated microbes for sustainable agriculture. So in today's talk, first I will tell you an important fungal disease of plants on which we are working. This is a very important disease of rice called rice sheath fly disease. You can see the rice stem being infected with this disease, and you can see the rotting of the stem. And this due to this, we loses almost 20 to 70% of the rice cultivation annually. And this disease is caused by a fungal pathogen called Rhizoctonia solani. So this Rhizoctonia solani will infect rice and will ultimately cause necrotic lesions, that spot-like, cell death-like of spots in the rice stem, and eventually it will kill the rice cultivation. So the source, generally the yield loss varies between 6 to 70% depending upon the environmental condition, and there is no resistant source which we can deploy it for the control of the disease. So people end up have to manage with fungicides. As I told you before that fungicides have residual effects. So controlling sheath fly disease remains a challenge. So our lab has identified a very novel bacteria from rice. This bacteria will live in rice, and when this bacteria, you grow it in a laboratory plate, it looks yellow in color. You can see this bacteria, which we identified as Burkholderia gladioli strain NGJ1. So this Burkholderia is yellow in color, lives in plants. And this is the Rhizoctonia solani, which causes sheath fly disease in rice. And when we observe that when this bacteria interact with Rhizoctonia solani, initially it prevents the growth of the Rhizoctonia solani. So you can see the zone of inhibition surrounding the bacterial colonies. So this shows that the bacterium has antifungal activity on Rhizoctonia solani. And interestingly, we observed that it also has a very novel property to feed on fungi. You can see in this plate, the fungus is almost eaten away by the fungi. So this bacteria has an unusual property to feed on fungi and use it as a nutrient source. So what it does, it will kill the Rhizoctonia solani and utilizes this fungal biomass for its growth. So this is a very interesting property of a bacterium, which is called microphagy. So myco means fungus, phagy means eating. The fungal eating bacteria we identified in our lab. So now the question was how this bacteria eats fungi. So we started investigating how the bacteria eats fungi. So this is a microscopic image. For many of you who are undergraduates may have difficulty understanding, but I'm trying to simplify it, that what this bacteria will do. This bacteria, the fungus is a myceliated structure. The fungus is a eukaryotic organism. So it is a multicellular organism. You can see the myceliated structure of the Rhizoctonia solani. And these spores are bacteria. So bacteria is a prokaryotic organism and fungus is a multicellular organism. So somehow we figured out this bacterium has an ability to attach it to the fungal mycelia, and subsequently it will start chewing away the fungal mycelia and eventually eat away the fungal hyphae. So you can see during initial stage of infection, the fungal hyphae is in the intact shape, but during 72 hours of interaction, you see the fungal hyphae is totally disintegrated. So this bacteria has lyzed the fungal mycelia and released the fungal nutrients into its extracellular environment, so that now the bacteria can use it. So we further demonstrated that indeed the bacterial treated Rhizoctonia solani is dead. So here I am showing it a particular assay called MTT staining. So MTT is a dye which is metabolized by a living cell. So you know the living organism will have active metabolism. So due to that active metabolism, the Rhizotonia solani will metabolize MT2 into a colored compound called formazole. So live Rhizotonia solani, you can see the brown, the appearance of the dark color. So this shows the Rhizotonia solani can metabolize MTT, it's living. But the bacterial treated Rhizotonia solani, you see, there is no formation of formazole, there is no formation of the colored compound. So this further demonstrated that the bacteria kills the Rhizotonia solani. So the take-home message is that this Burkholderia bacteria has an unusual property to feed on fungi. It disintegrates the fungal hyphae and kills it. You can see it here more apparently that without bacterial treatment, the Rhizotonia solani will grow in a cottony mass and you will have a ball, bigger ball produced. But upon bacterial treatment, the ball is shrunken and very significant. You can see how different it is. That here is a bigger ball, here is a smaller ball. So this again reinforced that the bacteria has eaten away the fungal mycelia and prevented its further growth. And also we figured out that the bacterial growth is manifold enhanced in presence of the fungal hyphae. So here is the bacterial growth in the rich media without fungal hyphae. You can see the bacterial is growing to the limited extent, but in presence of fungal hyphae, the bacterial growth is manifold enhanced. So all these two data emphasize that NGJ1, this Burkholderia gladuli, which we isolated from healthy rice seedling, has an ability to damage fungal hyphae and utilizes fungal biomass as source of energy. So we further tested whether this ability is restricted to one fungal or it can be similarly observed on many fungal species. And sure enough, we observed that this microphagous ability is broad spectrum. That means you don't only see it on Rhizotonia solani, but there are many other fungal pathogens for which this Burkholderia will eat them. So this broad NGJ1 shows broad spectrum microphagous ability. So now question is how to understand that bacteria is able to feed fungi. How the bacteria is able to feed fungi is a question. So to order to identify such biological insightful questions, what we do transcriptome analysis. So I'm sure you all might have read about it, that the function of the organism is regulated by genes and proteins. And so if we know how these genes or proteins are differentially regulated during particular treatment, we may be able to understand or identify certain important molecules or processes that may be playing key role during that particular process. So in this case, we were interested how this NGJ1 bacteria can eat fungi, can kill fungi. So we did transcriptome analysis. Transcriptome analysis will tell us that what all are the genes that are differentially regulated during that treatment. So we did bacterial fungal interaction transcriptome analysis and we identified genes that are upregulated during bacterial infection. And we observed that there are certain bacterial genes which encode a secretion system that are upregulated during microphagous interaction. So you can see this secretion system is called type 3 secretion system. So this is a multicellular machine, macromolecular machine, which bacterium uses to puncture into the host bacteria, plant or animals, and then inject certain proteins directly into the eukaryotic host. So for example, this is, you can see the bacterial membrane. You will have outer membrane, inner membrane, and peptidoglycan. So using this syringe, the bacteria will take its virulence factors and directly make a needle into the host membrane and deliver those virulence factor into the host membrane cytoplasm. So this is a very interesting way of bacterial delivery system. So people have figured out that this type 3 secretion system has role during bacterial pathogenesis in plants and animals. You might have read that we get E. coli infection, salmonella infection, typhoid, and things like that. So for those infection process, the bacteria is known to use this type 3 secretion system. Similarly, the bacterial pathogens of plants uses type 3 secretion system to establish disease in plants. And here we were observing that this Burkholderia bacteria is trying to upregulate type 3 secretion system to kill fungi. So this was a very interesting observation which we figured out in our lab. So people have shown that T3SS is important for bacterial pathogenesis in plants and animals, but people have not shown before that this T3SS is involved in bacterial fungal interaction. So we try to investigate whether indeed T3SS is involved in bacterial fungal interaction or not. For that, we now make mutation in the gene. That means we create a bacterial variant or mutant wherein that particular gene is disrupted. So if you knock down one gene, and if that gene is important for function, the bacteria will be not able to do that function. So this is called bacterial genetics and bacterial mutagenesis. So we mutated one of the key gene in type 3 secretion system, apparatus formation, and tested whether that gene mutant is defective or proficient in bacterial microphagy. So what we observed that when we disrupted type 3 secretion system gene in Burkholderia, the mutant bacterium is unable to forage over the fungi. You can see the mutant bacterium is only living here and rest of the plate is fungus grown. So that shows that the type 3 mutant bacterium of NGJ1 is unable to forage over fungi, while the wild type bacterium has completely prevented the fungus growth and has totally eaten away the fungi. So this clearly demonstrated that the type 3 secretion system is important for microphagy. So this was a very novel observation which we could unravel. So now the next question is, you might remember I told you that bacteria uses type 3 secretion system to send some protein from bacterial cell into the eukaryotic host cell. So we try to identify what all are those proteins. So for this, we did the genome sequencing of this bacteria. For those who may not know the genome sequencing, we can discuss at the end. But with this approach, we can be able to know what all are the genes that are being encoded by the bacterium or particular organism. So we identified the Burkholderia has around 5,000 such genes which is encoded in this genome, and amongst them we identified around 35 putative type 3 secretion system apparatus, and that we do using computational analysis. There is an important area of research that is called bioinformatics or computational biology. So you can use algorithms and you can write scripts to figure out what all are other genes involving in that process. So using that algorithm, we could figure out that the Burkholderia has 35 potential T3SS effectors. And one of them was showing homology to a prophase-like protein. So you know bacteriophage. So bacteriophages, bacterial predators, which can kill the bacteria. But what we were figuring out that the Burkholderia glanduli encodes a prophase-like protein as a T3SS effector. So this is very interesting why a bacterial predator, why an enemy of bacteria can be used as its friends to kill the other bacteria, other fungi. So that was very interesting. So we first demonstrated that indeed this Burkholderia glanduli prophase-like protein, which we named it BG9562, is a bona fide T3SS effector, which is delivered by the bacterium into the fungal membrane. And here is the data to show that. But I'm not, for technical detail, I'm not going to go into it. But if you are interested, I can discuss. So take it from me that this 9562 is indeed delivered by the bacterium into the eukaryotic host, into the fungal hyphae. So we further try to mutate this gene. I told you I introduced bacterial genetics approach. So we can again mutate any gene of interest. We mutated now, in this case, 9562, and observe that the wild type bacterium, where the no gene is disrupted, it is completely eating the fungi, while the mutant bacterium is defective in eating fungi. You can see the bacteria is only yellow bacteria, only at the center, rest of the plate is fungi. So that shows this mutant is defective in eating fungi, while the complemented strain is ability to kill the fungi or eat away the fungi, similar to that of wild type bacterium. So this shows that 9562 is indeed required for bacterial microphagy. So now question is, how a prophase-like protein is involved in microphagy? No report suggested that prophase-like protein can be involved in killing fungi. So we thought that maybe it may have some toxic domain or some lytic enzyme so that it can degrade the fungal cell membrane or cell wall and eat the fungi, kill the fungi. But to our surprise, there was no toxic or enzymatic domain present in this 9562 protein. So this shows that the bacteria uses a bacteriophage protein, but the protein does not have any lytic or enzymatic functions. So how the protein kills the fungi is not known. So what we observed that this Burkholderia prophase-like protein is present in many other bacteria also, and the sequences are quite similar. This is the amino acid sequence which will lead to the protein, and you can see the star shows the sequence conservation. So the proteins are quite conserved in diverse bacteria, but at the N-terminal region, at the beginning of the protein, there are certain amino acids that are only conserved in Burkholderia strain bacteria, but not in other bacteria. So this 9 amino acid stretch, we figured out that this is important for delivery. So we could demonstrate that 9 amino acid is important for T3SS mediated delivery of the protein. And when we complement the 9562 protein with the deleted variant, it's not able to complement, but with the full length variant, it is able to complement. Again, this is a little complicated technical slide, but just take it from me that the message is that this 9562 protein has to be delivered into the fungal membrane for antifungal activity. So also when we take this protein and now express in another bacteria, and if that bacteria has type 3 secretion system, that bacteria gains microphagous ability. So this we demonstrated using another bacteria called Ralstonia solanacearum, and our data show that if we heterologously express this protein, it will impart Ralstonia the ability to forage over the fungi. So the question is how 9562 is able to eat fungi, help the bacterium to eat fungi is not known. So we had the purified protein. So we started testing whether the purified protein can directly kill the fungi. So we observed, sure enough, that treatment with the purified protein itself is able to completely kill the Rhizoctonia solani. This is the protein treated Rhizoctonia solani. You can see that there is no growth of the Rhizoctonia, while the buffer treated one, the Rhizoctonia covers the entire plate. And also through microscopic analysis, we figured out the protein treatment, this to the disintegration and deformation in the fungal hyphae, while the buffer treated Rhizoctonia solani is completely normal. So this suggests that we can take the protein and use it for treatment, and that treatment can prevent the fungal growth. So of Rhizoctonia solani. So again, we tested that whether this protein can completely kill the other fungi also, and we observed indeed the 9562 protein shows broad spectrum antifungal activity. It can kill many pathogens, for example, Magnaportha, which is a rice pathogen. This is a protein treated one. This is a buffer treated one. And also, not only plant pathogen, it can kill the animal pathogens also. For example, Candida, it's an opportunistic human pathogen, and you can see a clear zone of inhibition surrounded upon BG9562 treatment. So this shows that the BG9562 protein has broad spectrum antifungal activity, but it does not have any antibacterial activity. We tested against many bacteria, E. coli, Pentoa, Xanthomonas, and so many, and the protein does not show antibacterial activity. So we now are hypothesizing that considering the potent antifungal activity of 9562, this protein can be used for control of animal and plant diseases. So before I tell you the practical implication of this, I wish to emphasize the biological insight which we have uncovered till now. So I told you the fungus is a multicellular organism, and this bacteria is a unicellular organism. So this bacteria has affinity to interact with the fungus, and during interaction, it upregulates its type 3 secretion system, which is a needle-like apparatus. And now this type 3 secretion system will help the bacterium to deliver effector protein into the fungal hyphae, and this effector protein subsequently will lyse the mycelia, and so that the fungal metabolites will come out into the hyphal cells, and that bacteria uses as a nutrient source to survive. So this is the insight which we covered during our study and published a very important paper in the field in Nature Communication, which is a very high impact journal of international repute. So if you are interested to read more about it, please go to this, and it is freely available, and read more. So till now, I told you that we have identified a protein which has strong antifungal activity. So now we are trying to use this protein for disease control. So we have obtained a variant of this protein and have patented this protein in various countries, India, USA, China, Indonesia, Australia, and Vietnam. And I have showing that if we just spread this protein solution onto the plant, you will be able to control disease. So this is the buffer spread plant infected with Rhizotonia solani. You can see the necrotic spores, which symbolizes the Rhizotonia infection. But upon protein treatment, you see the symptoms are significantly reduced, and only you see a little spores. And ultimately, there is very little ill reduction. So this shows that the protein treatment is capable enough for protecting against heat blight disease. And also we show that the upon protein treatment, the fungal colonization is significantly controlled by mere spray of the protein. So this is the buffer treated rice leaves. You can see the Rhizotonia solani growing extensively, colonizing into the rice tissues, while upon protein treatment, you hardly see any fungal growth. So this clearly demonstrates that this protein treatment can be potent enough to control the disease. So we have also tried to spray this protein under field condition, and we observe indeed the protein treatment is protecting. So now we are collaborating with another ICR institute, which is agricultural organization, and we are trying to develop a protein-based formulation for disease control, and we are at the field testing stage. Another way of using the protein for disease control is through transgenics. So in transgenic, what we do, we take the gene and we try to express it in plant now. And we can take the gene, integrate into the plant genome, and now the plant will continuously produce that gene or protein, and then whenever the pathogen will come, it will kill the pathogen. And we indeed observe that when we heterologously expressed in rice, we observe a significant tolerance against heat blight disease. So here you can see this is the control rice plant which is infected with Rhizotonia solani, and the entire plant is dead. While this protein overexpressing plant, you see they are healthy, and even you can see the fruit set. So we have clear demonstration that the overexpression of 9,5-CBG, 9,562 protein provides disease tolerance against Rhizotonia solani, and also we are coming up that those transgenic plants have multiple fungal disease tolerance. So we told you that the 9,562, a single protein of the bacteria, is potent enough to protect a important fungal disease for which we don't have disease control. And so now I will shift the gear and talk to you about some antibacterial activity of the protein. So what we observed that this NGJ1 bacteria, which I have been telling you that has strong antifungal activity, we observed that this bacterium also has strong antibacterial activity. You can see the zone of inhibition for different bacteria, which symbolizes its antibacterial ability. And we observe that this NGJ1 again has a broad spectrum antifungal activity. And we have figured out that bacteria uses different secretion system to kill the fungi. So it has two different secretion systems, which we are referring it as T6SS1 and T6SS2. So using this bow, it will deliver various toxin proteins into the another bacteria and kill them. So this mechanism we have just published in EMBO report, which is again a very prestigious journal. And again, this is freely available. So if you are interested, you can read this detail. But the take-home message is that the single rice-associated bacterium has potent, very strong antifungal activity, very strong antibacterial activity, and those bacterial and antifungal activity has practical implication in disease control. So now I try to, so until now, if you have any questions, we can break for 2 minutes and discuss the questions. If not, then I can proceed further. So if anyone of you are interested, please let me know.
Sir?
Yeah, please.
First of all, your talk has been very interesting so far. The thing that I wanted to ask was, you said that the protein that you isolated has also acted against Candida and other human pathogens. So
Yes.
is there potential to actually use this for treating fungal diseases in humans?
Yes. That's what we are developing now. We are trying to explore whether this protein can be used to control fungal infections in animals.
Okay, sir. And the other thing was you identified that 9 amino acid sequence was very important for delivery of this toxin.
Yes.
So did you try creating a mutant without that 9 amino acid sequence to confirm the activity?
Yes. I did that. Actually, the slide which I skipped was like that. I will come to that and tell you. I thought that maybe many undergraduates may not understand. So that's why I did not go into the detail of it, but I will take 1 minute and explain it to you. Give me 1 minute. Yeah. So what I did is, I told you that this 9562 protein is the full-length protein, and you can put it in a plasmid which can express in bacteria. And again, so this is the full-length protein cloned in a PHM1 plasmid which can replicate in Burkholderia. Now this is a deleted variant where that 9 amino acid, that means 27 base pair is deleted. So this deleted variant, we cloned it in PHM1. So now the full-length protein clone, I'm referring it as PGD3, and the deleted variant clone, I'm referring it as PGD5. So now this is the wild type where it shows the microphagy. You can see the bacteria growing over the fungi. This is the mutant bacterium. I told you that mutant bacterium will not grow. Rhizotonia solani only growing. This mutant bacterium where full-length protein is expressed with PGD3, you see it is microphagy. So that means the expression of the full-length gene is able to restore microphagy. And this is the mutant bacterium with PGD5 where 9 amino acids is deleted. Now this mutant bacterium is not able to forage over the fungi. So that clearly demonstrates that the secretion signal and T3SS mediated delivery is important for antifungal activity. Thank you. So hope it is clear.
Yes, sir. And the sequence is missing in pathogens like Burkholderia cepacia, which is found in humans, right?
Yes.
Oh, okay. So I was just wondering if it was somehow a protective mechanism in that organism when it infects the humans.
Yes. Good question. Thank you.
Thank you, sir.
Thank you. Any other questions? Okay. So I will now move further. So good that some of you have at least followed, and we expect more questions at the end of it. So now the take-home message I gave you, that I showed you the potential of one single organism, one single bacteria, which we isolated from plants, rice, and showed you that this bacterium can be potent enough to control fungal and bacterial infection. But now I am trying to emphasize it, that there are large number of microbes, millions of microbes live within or in association with plant and in that sect in animals. And they have profound impact on the health, growth and development of plants and animals. So can we harness those microbes for sustainable agriculture is the question. And I am going to tell you briefly that yes, we can harness that. So to re-emphasize the fact that large number of microbes are living in plants, so you can see that there are lots of microbes which will live in the rhizosphere. Then there are microbes which will be intracellular. There are microbes which will be endophytic. There are microbes which will be externally associated. So almost all plants of the plants have microbes. So there can be air microbiome. There can be phyllosphere, that is the aerial part of the plant microbiome. Then there can be endophytic microbiome that lives within root, stem, leaf, seeds, pollen, etc. And then there can be rhizosporic microbes that is living in association with the roots of the plants. And then there can be the soil microbiome. They are closely associated with the plants. So these plant microbiomes, you can imagine there will be millions and millions of microorganisms which live in plants, and they are living organisms. So they will not live alone, means isolated, inert, inactive. No, they all are biologically active, and they all have their genome. So imagine by birth, we are born with the same genetic content which we inherit from our parents. But these microbes, which we acquire during our lifetime, adds up new genetic sources into our microbes. So this is our extended genome, and those genomes help us to combat many important events in our lifespan. Similarly for plants. So I'm telling you that there are many microbes which has been isolated from plants, and they are having important aspects. So for example, there is one microbe which is Bacillus. I told you example of Burkholderia. Similarly, Bacillus strain has been widely characterized in literature, and the strain has been found to have antifungal activity, antibacterial activity. It has ability to modulate the biofilm formation and bacterial motility, and also the Bacillus can be elicitor for ISR activation. So there are many facet role of Bacillus in plant control. So I can extrapolate it in a bigger term, these Bacillus are the plant-associated bacteria, can be formulated as biopesticides, they can be biofungicides, they can be biofertilizers. So this bacterium can have multifaceted functions, and they are biologicals. So they are not chemicals. So they are biologicals, and they will not supposed to have adverse side effects if we have characterized in detail. So they are biologicals, and they can be extensively used for control of the disease. So people are trying to identify synthetic communities. So they will isolate a good number of microbes which are having beneficial property. I'm not telling that all the microbes will have beneficial property. Many of them may be opportunistic pathogen. Many of them will have allergens activity and things like that. But at least good handful of them will have beneficial activity, and we can isolate them and make a synthetic community. So SynCom of the beneficial microbes is extremely useful for beneficial activity. And here I am showing you that people have figured out a synthetic community consortium which can enhance the plant growth also. So this is the control tomato plants growing happily. But if we treat the plants with SynCom community, the growth is significantly enhanced. So this shows the bacterial utility are the consortium of the microbes promoting the plant growth as well. So now people are trying to have a smart way of designing SynCom, because earlier isolating one bacteria, characterizing them, adding them will really take lots of time, and the bacteria function in community. So if you take one bacteria, may not be growing to the same extent in the field condition and in the natural condition. So if we can keep the consortium and then you can put all of them as a friends to the plants, it grows well. But initially people were trying to characterize one. But now we have genome information and we have computational tools. So we can use the culture collections, genome informations, and then using bioinformatics tools, computational tools, we can predict who all can fix well, who all can grow together, who all cannot grow together, and calculate what all are the synthetic community which can be stably inherited in the plants. And then synthetic community, members of those community can be isolated and can be used for stable crop cultivation and things like that. So with this, I end to my talk, and I'm thankful to my students who have done great work and have been always hardworking. And also I've been generously funded by Department of Biotechnology, Department of Science and Technology, and NIPGR and Swan Jayanti Fellowship. Thank you. So I kept good time for discussion. So let's discuss if we have questions.
Thank you, Doctor. The session is open for discussion. If you have any questions or need clarifications, you can ask Dr.
And even technical details, I'm sure many of you may be willing to understand what is genome, what is transcriptome, all those things also we can be able to address. One of you have been asking the link of the paper. So PPT slides I will share with Professor, and he can share with you. And the link of the paper, if you just put the title of the paper, you can be able to download it. And you can just put my name in Google. You will get lots of papers, and then the relevant papers, you can download it. They all are freely accessible.
Any questions?
So you all are students or teachers? They are students. Huh?
Undergraduate students of life science.
Very good, very good. So clear what I tried to show you, opened up a interesting biological questions and showed you how we can do detailed research study and try to identify something very unique and how to use it for disease control. So that is interesting. I think they don't have any question. Yeah. So in case you have any queries, you can request Professor to pass on to me, or you can write to me also. You can write down my email ID, jmsgopal@NIPGR, National Institute of Plant Genome Research.ac.in.
அப்பதானே அவனுக்கு தெரியும்.
In case of no questions, I would like to thank Dr. Gopalji for joining us today. Thank you so much, Doctor.
Yeah, my email ID is wrongly written, so I'm just typing it. Okay. So thank you so much.
Thank you so much, sir.
Yeah, thank you. Nice meeting you. Great. All the best. Thank you, Daksha. Bye. Bye. I'm logging up.
Okay.